Hopefully we will all get to go outside later. So I will welcome all of our speakers back, our panelists back for this panel discussion, which I think is going to be a good one. And we have quite a number of questions. We have 14 questions lined up. So I don't know if we'll get to all of them, but we will certainly do our best. The first one, Scott, we're going to start with you, but then I'll open it up to Yvonne and Chris as well. Direct-to-consumer fecal testing is variable, as you described. But what about results from a medical professional to a patient? Are there highly controlled clinical microbiome tests at this point?
No. Again, it goes back to the methodological variability, and I can comfortably say that every microbiome measurement that's ever been done is wrong. It's inaccurate. The saving grace in all of this space is that they're precise, they're reproducible. So differences that you see are real. So if you look at cohort of vegans versus omnivores, you see differences. Those differences are real. You might be measuring them wrong individually, but the relative differences are real. So the reports you get from your microbiome analysis typically show where you are relative to a cohort or a population, a self-reported healthy population. So, here's the cluster of healthy people and you're right here, So that's about the best we can do right now. And even if you could do absolutely accurate measurements of the microbiome, there's no way to really interpret that. It is all relative based on like, where are you in a healthy versus disease cohort? That's about the best we can do right now.
Chris, are you on?
I am, and I completely agree. I also think there are a couple companies out there that have linked those relative differences to things like blood sugar. Yeah. And if there are any, and I say that with a question, companies that are doing this in a meaningful way that provides guidance clinically, it would possibly be those companies, because what you do is you send in a stool sample, they perform an analysis of it. They know that certain types of microbes and genes are associated with certain responses to foods. And so they can tell you, well, you can eat this food and you'll respond just fine, but you don't eat this food because it's going to spike your blood sugar. That's actually been studied pretty well, and it seems to be pretty predictive. It's just not everyone needs their blood sugar controlled like that. That's more relevant for folks that have diabetes. If you just want to know what your microbiome is, you could do that for kicks and giggles. It's just not going to be terribly helpful.
Yeah. Yeah, and to add to that, I think Scott and Chris have summarized this beautifully. It's the equivalent of asking if we have true north measurements of the microbiome, which Scott's earlier presentation covered this really well. We do not, right? We can't accurately say that what we are seeing in the fecal sample is an full accurate representation of what is inside of your body, right? That's a very difficult assumption to jump to, or conclusion to jump to. However, there are ways to control the process and the measurements, which is what Scott's group is working on heavily. I do as part of my job as well. A lot of it has to do with making sure we're standardizing the collection method, the storage method, the transportation, the actual processing of the sample, and making sure that the data analysis we perform is reproducible, right? Even if we don't have a true north accurate representation, we can at least make sure that we are measuring the same thing the same way every single time. And so once we do arrive at that point, if a company or individual group is able to do this, they can take this to FDA, go through kind of like a de novo pathway, get a full kind of path or a full kit approved pretty much. And we do have a kit approved for collecting and storing fecal samples for microbiome analysis by the FDA using this pathway, but the actual downstream analysis portion is still lacking these tools. So it will take some time to get there, but I think we are working towards that.
Wonderful. The only thing I have to add is something that I say to my students all the time. Would the test change what you were going to tell them to do? right? If you were going to tell them to, have mostly whole foods, increase their fiber intake, get more physical activity, regardless of what the microbiome test says, then do you really need a clinical microbiome test? I would argue probably not, at least not at this point, because we just don't know enough to get any more granular than that anyway. So wonderful. Thank you all.
Francis Collins, the NIH director, of course, had a great saying. He was asked a question during a seminar. A person asked, should I get my genome sequenced? And Francis Collins' response was, use that money to join a gym.
That's a great response.
Yeah.
Yeah, same thing. Save your microbiome money and use it for buying healthier food and getting some physical activity.
Yeah.
Wonderful. Thank you. The next question, I'm going to have Yvonne start with this one, and I think Scott might also have some thoughts. Is diversity modified with age when evaluated with shotgun metagenomics or other more advanced methods, i.e. transcriptomics, as well as the more typical 16S metagenomics? Basically, is there a difference with these new techniques?
So we do see that we see increasing amounts of information collected basically with the different methods, right? 16S, you're really just at the surface, and then you begin to dig in with metagenomic shotgun, and you're getting more information, more data. Then you add metatranscriptomics and meta, basically just a lot of information start coming through, right? And so I will say that increasing diversity over the lifespan, especially within the first, I want to say 20, well, about 15 years of life, really. By age 1, as I mentioned, your microbiome is fully pretty much established, right? But that diversity can go up based on diet. It can also go down. Right? Let's acknowledge that, again, as Leigh has covered in her lovely presentation. So I think that regardless of the method that we use, all of these studies have the same conclusion. Diversity does change based on the age, but a lot of it is actually dependent on diet and lifestyle. And maybe Scott or Chris want to add something?
Leigh, you cut out when you were asking the question, but I just think it's whole genome shotgun versus 16S analysis. And
and also, other methods like transcriptomics. Are we seeing a different picture with them?
I will say that a different picture in terms of, I guess, just the type of information being collected, right? So I think that's what the question is about. Are we getting a different type of information collected? Yes. But in terms of the actual core question of if the diversity itself is being modified over your lifspan, yes, we still see that play out regardless of the technique you're using to collect that information. So if I was looking at just, for example, 16S, right, we can answer that question, just look at genera or like a genus, maybe species level, probably not. With high reliability accuracy, but at least genus level, I can say that, these genera, these phyla are affected this way over the person's lifespan. And I think I see a chat said regarding diversity specifically. Yeah. So that's going to be regarding diversity specifically if we see that pattern play out regardless of the method. And I think, yes, based on what I have seen, we do see that regardless of what method or technique is being used to collect the data. That is what the data suggests.
Yeah, that sounds right on. And what I might add is the taxonomic, or the players that are there, is going to be more or less similar. The functional piece, which metagenomics and transcriptomics is getting at, what's really interesting is the diversity as you get older gets greater and greater. But the functional piece actually maintains more constancy. And that's because the functional part of our genes is incredibly redundant. So you're getting more and more players that have those same types of genes. And that's what the metagenomics and the transcriptomics is capturing, is that functional part. Like what are those bugs actually doing there? What does that mean? How does that reduced down to practical implications of how we live our lives? I don't know, but it's an interesting finding.
Yeah, and to add to that from a microbial ecology perspective, right? If we look at the gut microbiome just as an ecological community, and we think about each of the different groups of microbes we see there mostly as just a functional entity, right? Filling a specific ecological niche, right? Typically what we see is that there's, just like Chris mentioned, there's redundancy built in there. So there's multiple groups of microbes that perform exactly the same function. So short chain fatty acid producers like butyric producers belong to multiple genera. It isn't just limited to one specific organism. So even if that organism is lost in the microbiome early in life, there is another one that fills that ecological niche.
Mm-hmm.
All right, I think we covered that one. Next question is a simple question, but I don't think it has a simple answer. Does a quality probiotic need refrigeration? Who wants to tackle that one?
Well, remember that foods are also categorized as probiotics, fermented foods. So, are you talking about kimchi? Are you talking about yogurt, sauerkraut? Or are you talking about the bottles of pills that you buy from CVS?
I think they're probably talking about the bottles, but I think you're fair to include kimchi and yogurt and all that in there.
So from a manufacturing technical perspective, they are required to write the number of viable bacteria on the side of the bottle, and it's usually like 10 to the 9th live cultures or something. They have a weird terminology, but basically it's colony forming units. And what the manufacturers do is safety. If they say it's 10 to the 9th, it has to be 10 to the 9th 2 years out. So what they'll do is they'll do an overage, like an order of magnitude or 2 orders of magnitude over, so that when 2 years comes along, it's been reduced, but now it's back to 10 to the 9th. And that's for what it's worth, it's just how they handle that. Because yes, the bacteria will die slowly over time. For refrigeration, will probably slow that down. But we're talking about logs, orders of magnitude of colony forming units here. So, a twofold decrease is insignificant in the scale of 10 to the 9th.
That's brilliant. And what I might add is there's some really fascinating research that's been published in the last 5+ years that looks at live bacteria versus killed bacteria, live inactivated bacteria. I think there's one study done for Akkermansia in particular in metabolic disease, and if I recall, there wasn't a difference for that particular study in outcomes, which says that it's the preformed components of that Akkermansia that was responsible for what the effect was. Now, I don't think that categorically is the case across all probiotics. It really depends on the specific probiotic, and I think this was your point, Scott, earlier, and B, the specific indication. So we can't say categorically that you need live bugs or dead bugs, but that possibility that dead bugs work and data that supports dead bugs work, I find intriguing.
Yeah, and some of the interesting thing about that is it the dead bugs or what comes with the dead bugs, right? The postbiotics, is that what's actually working?
Yeah, exactly. I actually unmuted myself to say what Chris said, so I'm just not gonna say more. You summarized it really well.
Yeah, you did summarize that really well, Chris. Actually, the next question is for you as well, Chris, but I think we'll all have thoughts on it. Is regular use of a prescription antacid a concern for the gut and its microbes? PPIs.
Do you want me to go first, or?
Yes, please.
Okay.
Start with the gastro.
There's a ton of research coming out on PPIs and how maybe we shouldn't be adding them to the water like fluoride. And prescribing them, so readily as gastroenterologists and as internists, et cetera. Part of that research is actually around the microbiome and the impact that antacids have on the microbiome. That truly just about any medication that you take has been shown, even, drugs for depression or for psychosis have been shown to impact the microbiome. But yes, acid suppression medications do as well, for better or for worse. And anytime you prescribe a drug, it's a risk-benefit analysis. And In some cases, PPIs, you know, have incredible improvement on quality of life for people, and they're valuable. Yes, knowing that there are these side effects. But the answer is, yeah, like they can impact microbiome by decreasing acid or having direct effects lower on in the gut.
That's an application that we've discussed for our NIST standardized fecal material, that you could use it to do some in vitro drug interaction studies. You know, you add a PPI to an in vitro, you know, fecal sample and let it go and see what happens as sort of a, you know, an in vitro model system to understand those processes. And the fact that it's standardized and we can distribute it across the country means that people can reproduce those across laboratories.
So I just see a comment in the chat from one of my faculty members reading my mind. So Mark Webster, he says, PPIs are my least favorite drugs because they also impact nutrient absorption as well as reduce the acid that would normally kill oral bugs that enter the stomach. And I would say the nutrient absorption is a concern that I have as well, particularly for magnesium, which most people don't get enough of it already. So now we're layering that on top of this. And I think that is often not included in the cost benefit analysis when we're thinking about these drugs. And so doing a more holistic examination of that cost benefit analysis might shift how many people are getting these drugs.
I mean, this may in truth be part of the mechanism behind the bone density findings that are seen, you know, impact in divalent cations, specifically calcium. And other divalent cations actually help in calcium absorption. Along the lines of fiber, though, I think a story that we'll see developing in the future, there's already some evidence to support this, is fiber is not relevant just to lower gut. And it may or may not be through a microbiome mechanism that it impacts things like not the actual reflux, gastroesophageal reflux disease, but how the esophagus experiences that reflux, whether the barrier is broken, whether those nerve endings are potentiated and inflamed and, you know, scream when they feel that acid. Yeah, there's even commercial products out there that have evidence behind them, I think out of Stanford, that show an improvement in heartburn symptoms. And it would be great. I'd love it if we could replace PPIs for all the reasons that you just mentioned, Leigh, with these new fiber-based and whole food approaches.
I have
Amen to that.
Yeah, I have a family history of acid reflux, and I used to take PPIs and needed to because of acid reflux, but through some lifestyle changes, I was able to stop, and I don't have acid reflux. So it's certainly something I think you can manage without PPIs.
That's the goal. So here's a question I feel like I get all the time. Can probiotic food be more beneficial or as beneficial as probiotic supplements? I don't know who wants to start with that one. Okay, I'm going to start actually. I'm going to take moderator privilege. So I think the main difference between probiotic foods and probiotic supplements is that probiotic foods are bringing so much else along with them, right? They have all these like things coming to the party with them, the prebiotic fibers, the non-prebiotic fibers, nutrients, the food matrix effect. There's probably some sort of consortia in that prebiotic food that maybe is not going to be present in a supplement. So my bias is that our body co-evolved with these fermented foods, and so there's probably a real benefit to having them in our life. That being said, the supplements are really helpful for precision and targeted interventions. And so I see that being their strength, but I don't know if we're really there yet.
You know, the health benefits of probiotics go back to fermented foods, right? You know, humans have been eating fermented foods for millennia. So this idea that you could take the microbes out of those fermented foods and it would have the same beneficial effect is kind of ridiculous to think about. There was a study done by a German group a few years ago, and they looked at 300 healthy individuals. And what they were looking for specifically is the presence of common probiotic strains in these healthy people who don't take probiotic supplements, and they didn't find any. So it seemed to demonstrate you don't need these probiotic strains to have a healthy, to be healthy.
Yeah,
Or perhaps they're transitory because, they didn't engraft.
Exactly. Yeah, I was going to add to that and say, really spot on with both Leigh and Scott's response. The food that you're taking is not just the bug itself. Some of them don't even have live, versions of the organisms that fermented the food. Some of them just have the remnants of that fermentation in it. And so when we take the bug alone by itself in a supplement, we're hoping that we can provide it with exactly the same conditions and exactly the same kind of substrate it needs in order to produce these beneficial byproducts that are present in the food in your body. And that's not necessarily what happens all the time, right? So I think really, again, this just points out that the reductionist approach to healthcare is not necessarily fully beneficial all the time. Again, it can be very helpful for targeted precision. So for example, you've had a health intervention that has really, really impacted your microbiome stability. And so you might want to take, some strains along with some dietary modifications to make sure those strains can then engraft because maybe you lost them through antibiotic consumption, right? Those are the types of things that then it makes sense, but on a routine basis, obviously, we're not doctors. We can't give you medical advice, except for Chris. He can, but he's not your doctor, right? So I don't know that he can either. But really important to note then that we are back to the whole foods, right? So just eat the whole foods, I think. If you need the probiotics on top of that, obviously go for it. But really important to that, just emphasize the need to have the whole food itself. So the fermented food is definitely getting the top vote from this panel.
The really cool thing about fermented foods, there's a study that was published out of Stanford, out of Justin's group, actually, Justin Sonnenberg. I think it was about 1.5 years, 2 years ago now. And he showed that increasing fermented foods in individuals increases diversity in their gut. And you think, okay, well, that makes sense. Fermented foods, lots of new bugs. What's crazy though, and counterintuitive, is the increase in diversity was not from the bacteria that was in those foods. So those fermented foods were like serving as almost like a prebiotic. They were promoting the growth of bugs that were probably already in the gut, just underrepresented. So super interesting. And so on target, everyone, like fermented foods over probiotics, albeit if people have a devastated gut microbiome, like a bleached coral reef, if you can imagine that sort of vivid image versus like, an aquarium type reef, it's going to be really hard even with fermented foods to restore that. And so, new therapeutics companies working on these approaches of using not probiotic food-based therapies, but the ones that actually are normally present in our gut to try to repopulate that. I think that type of approach for folks that have like true dysbiosis, true imbalance, is super promising. The folks that maybe normally can't have whole foods because their normal metabolic capacity is just devastated. It's not there anymore.
Very important point. So related to that, and Chris, we'll start with you on this one. Are prebiotics necessary for GI health versus probiotics alone? I think we already touched on that, but I feel like the necessary for GI health is important. And then a follow-up question, what fiber is best for GI health?
Yeah. Prebiotics are great for gut health. It's more than just fiber. It's the 4S fiber, phenols, ferments and good fats, I guess phonetic Fs because of the pH cheat. And then the second part of the question was?
What fiber is best for GI health?
What fiber? Diversity. So if you go for just all of them. If you go for a single fiber, you're very likely to miss the mark, and you're only going to grow one type of bug because of that one-to-one relationship in the primary degraders. So if you're doing a supplement, it's good to have a supplement that has diversity, but even better is to go for whole foods that inherently has that diversity built in.
Agreed. And Go ahead.
Yeah, the idea that probiotics are just sort of a marketing scheme to try to sell something that they can convince you is healthy for you because we've eaten them for millennia in fermented foods. I think we're about to see that again with prebiotics, which are fiber supplements. Oh, there's this new thing out there, prebiotics, and they're demonstrated to be healthy. These are also known as fiber supplements, and they've been sold for 100 years now. So it's just a new way to market something that's been around forever.
I think that's a fair point. I'm going to move on to the next question, which is directed towards me. Does drinking bone broth play a role in a healthy gut microbiome? I'm going to use this as an opportunity to discuss how the microbiome and food relate to each other. So when you digest a food, a large portion of that food is typically taken by you. You're like, hey, this is mine. Between the stomach and the small intestine, you absorb most of it. And so what the microbiome actually sees is what's left over. And what is typically left over is fiber, the things that are not digestible to us, resistant starch, things like that. Some amount of protein, which is going to vary based on your diet. So if you have a high protein diet, a lot of protein will make it to your gut microbiome. If you have a low protein diet, less protein will make it to your gut microbiome. And there shouldn't be much fat there if you have a healthy functioning gut and no malabsorption. And so if you're having something like bone broth, which is really minerals, vitamins, some fat, some amino acids, that's highly digestible. And so what happens is once it gets to your stomach and your small intestine, poof, it's all gone. There's nothing left, right? So it's going directly to you. And that's part of the reason people actually promote bone broth for health, because it is highly digestible. So even if you have a gut that's not maybe super functional, you can absorb the nutrients from that bone broth. But that means there's nothing left for the gut microbes. And so it's not for them, it's for you. We actually just had a post about this with my students in nutritional immunology, talking about fiber is really more for the microbiome than it is for you, but you need it because you need your microbiome. So let's take care of them. Any other thoughts on that?
Just one other thought, like I've become really intrigued by potassium recently and the relationship to potassium, the sodium potassium ratio. Most of potassium is absorbed, 90%, but there is a small fraction that makes it down. I don't know about sodium, I'd have to look that up. But as you're talking, it's kind of sparking this, all right, got to go Google it now, which is kind of my modus operandi. I'm always collecting questions. So thank you.
Yeah, that's a good question. And I think, so in the nutrition world, historically, we thought about digestion as stopping before the colon. And that digestion was just like where water got absorbed. And I think now that we understand the microbiome better, we realize that's probably not 100% accurate, but it hasn't been really well studied. So I don't even know if we know the answers to that quite yet. So much research to be done.
And I do see something in the comments about honey related to the discussion on fiber. So honey is a pretty rich source of a lot of these different complex oligos or carbs. So these are the fibers are complex carbohydrates. So there are some studies in animal models, in culture models in the lab, suggesting it has prebiotic effects. Not that many in human, and honestly, the ones in humans have not confirmed this. So, I mean, again, I don't know that it has, if it doesn't have a detrimental effect on your health, why not? But again, we're not here to give specific advice either way, just to share what the scientific evidence suggests. And so that's kind of the status of honey at this point, unknown, to be determined.
I'm glad you picked up on that one, because that's like a little pet project for me and for Misha Kogan. We're big fans of honey. And there is some human research indicating that small amounts of honey in the diet can actually improve your blood glucose control, which seems counterintuitive, right? You're adding sugar to the diet, but honey is so much more than sugar. And if you're only having like a teaspoon a day of honey, it's not a lot of sugar, but you're adding other things in. And one of the things that I like to think about honey, if it's raw and unfiltered, it's almost like a bee FMT, right? It's almost like I'm getting some of the bee fecal microbiome and transplanted into me. No science behind that, right? That's just my surmising. But I think that someone should do some honey studies. And actually, Dr. Kogan and I had tried to do a honey study, but we didn't get funded. So maybe in the future.
That sounds really cool, though. You could look at the the fecal microbiome of the honey and relate it, of the bees themselves related to the honey they produce, and see if you're actually doing a BFMT when you take honey. Really compelling, actually.
I think it's fascinating personally. Another thing I think that is very fascinating is intermittent fasting. And there's a question here about the evidence for that to reset the gut microbiome, which I think is a very specific question about how intermittent fasting might affect the gut microbiome. Who wants to take that one? It came in during your talk, Chris.
Okay. So generally speaking, timing of eating, I think, will be an increasing theme in metabolic control. Intermittent fasting is part of it. Not snacking between meals is part of it. The food that we eat is translated by the microbiome into signals that entrain our circadian rhythms almost as strongly as light cycles do. And when you travel and want to avoid jet lag, some of the best advice is to time your meals accordingly to the new time zone that you're going to help in that transition. So yes, I think the timing is very important for circadian rhythms, and circadian rhythms, furthermore, at least in animal model studies, with all the caveats that we talked about, has been tied to good metabolism and dysfunction metabolism when that circadian rhythm is disrupted. In other words, independent of increase in calorie intake, there may be a propensity for fat deposition and dysregulation in general if the timing of your meals isn't appropriate.
Yeah, I couldn't agree more. There's a whole field called chrononutrition about the importance of eating at certain times. And just for those at home, quick hack, if it's light out, you can eat. If it's dark out, don't eat, right? It's very simple. Like that's what the human body is used to, and it helps with things like sleep. And it's just, that's a pretty easy thing to do. And probably there's some sort of evolutionary biology behind that. And we've seen, there's been some studies looking at melatonin and other markers that if you eat at times, too late into the evening, your melatonin production is not what it needs to be. So you're not going to sleep as well, and then you aren't going to feel great. So these are all little small things you can do to hopefully improve your already good health.
Yeah, just to add a little bit on the literature end of things with fasting. So some of the literature suggests, there is some reshaping and the gut microbiome occurring with fasting, but it's important to note all of the disclaimers that everyone on this panel has shared with these studies before, but also to point out that these were extended period fasts. And so intermittent fasting is typically, sun up, sundown type fasting, or 16 hours fasting, 16 on, 8 hours eating, or 18-6, and that type of variation. Very few of those studies have been done. So most of the studies are extended period fasts of like 3 days or 5 days or whatever. And one of the really great ones came out of Germany just last, I think 2021. And they were looking at a hypertensive population and showing that a 5-day fast actually reset their gut microbiome and showed some pretty great beneficial effects there. So, it could be working through modulating the gut microbiome, intermittent fasting or fasting in general, but we definitely need more information on it, I think, more research.
Great, thank you. All right, next two questions are for you, Scott. The one is a quick technical question, which, and of course he's coughing, which you will have no problem answering quickly. And then the other one's a little more complicated. So the first question is, when diluting fecal sample with water, do you use standard sterile tap water or something special?
Just high grade molecular biology grade H2O. The higher percent H2O, the better.
Super pure.
Super pure.
Great. The next one is also related to your sample choices. They wanted to know why you chose vegan versus omnivore for your fecal standard cohort. And one proposed option was why not whole foods plant-based? Mm-hmm. And then related to that, do you standardize samples according to area of the country, demographics like ethnicity or type of diet?
Yeah, those are good questions, and we get asked that a lot. And it sounds ugly to say this, but we don't really care about the vegan and omnivore diets and their impact. We just wanted fecal standards that were different, something that we could say that this one is very different than this one, because it allows you to show and demonstrate what we call pseudo-quantitative differences. So I can't tell you what the level of E. coli is accurately in this one or this one, but I can tell you their relative difference. So it allows you to show quantitative relative measurements by having different compositions of materials. We decided on the diets because we theorized that they would have the largest impact on the fecal content. That's why we chose that. The second part of that question is, within the cohorts, yes, we age and demographically match them the best we can. They were all self-identified as healthy, and yeah.
Thank you.
And
Okay,
they were
Yvonne.
also screened for infectious diseases, hepatitis, HIV, things like that, because we don't want our stakeholders working with fecal material that has, infectious HIV in it, of course. So they were screened as healthy from infectious disease perspective as well.
Thank you. Yvonne, this question is, I don't think this is an easy one, but maybe I'm wrong. Do we know which organism or organisms in the gut make serotonin?
Oh, yeah, so not an easy question to answer. We have some ideas on which microbial groups or which bacterial groups are more likely to make serotonin. So that's the best way we can answer it, because we're doing this based on, predictive genetic sequencing and all of that. And that's challenging to demonstrate, like, in a very, very direct way, especially inside of a body. But there have been some animal studies trying to take that genetic information to prove that these are the specific organisms that make serotonin. So there are some that are suspected, but at this point, I will say it's still really early and hard to be confident that those are the true north answers. They're exactly right. I think we have to take all of that with a grain of salt at this point, and then wait for more research to come out.
Yes, agreed. That makes sense to me. All right, next question. This came in during your talk, Yvonne, but I think anyone can see if they've heard about this. Have there been any studies that you are aware of using FMT for infants born via C-section after delivery from their mothers? I can't think of any.
I can't either. Now I'm like Chris, I Google everything. Now I'm gonna have to go search PubMed and see if I can find something. Oh, clinical trial database.
Yeah, Scott, did you see that when you were looking at the clinical trials database?
What was the question again?
Infants born via C-section, are they using FMTs to try and improve their gut microbiome because they were born via C-section?
No, they wouldn't do that to an infant because of the risks associated with it. There are probiotic strains, namely Bifidobacterium longum, also known as baby biff, and that has been used and approved for neonates because it's known to be beneficial to infants, namely through human milk energy absorption. FMTs are risky. FMTs are risky, and you don't want to use it willy-nilly. So
I did find one Nature paper actually a couple years ago. They used the mother's fecal material, so just to improve that initial seeding of the infant's gut. I think they took it directly from the mother and gave it. It was just a proof of concept though, which basically means it was a very small set of babies.
Yvonne, your comment, Rob Knight's book, Follow Your Gut, Rob Knight is a leader in the field, and in his book, he talks about his wife giving birth via C-section, and he inoculated his baby in the delivery room.
Oh, he did the FMT.
Of course he did.
Yeah, and he said the nurses and doctors were looking at him like he was crazy, and he was trying to explain to him what he was doing. So,
but that was with a vaginal swab, right?
He was using vaginal swabs. Yes.
Yeah, which is a little bit less risky in theory.
Mm-hmm.
Yeah, and this was done orally, so it's interesting. They took the poop from the moms and mixed it up and gave it to the babies orally. Yeah.
And what year was this in?
It's like 2020.
Okay, so there could be
It came
follow-ups
out.
coming.
Yeah, they did. They just showed data for 3 months of follow-up.
Interesting.
No adverse events reported. So
I've heard conflicting reports, different papers that support C-section as a different microbiome versus vaginal birth. In the latest report that I saw, there was no difference. One of the theories is that it's actually breast milk that helps seed the gut, and that all of the microbes that are necessary are already there. And there's even theories that there's some crazy like gut breast connection that allows those microbes to make their way into mom's milk and then out to the baby. That also is controversial, but I find that intriguing.
Yes, that sounds very controversial, but intriguing. You're right. Like I want to see more on that. All right. So we have a few questions left. Chris, I'm going to send this one to you. Why do some medical doctors, including gastroenterologists, not believe in studying the microbiome? I had one gastro tell me that probiotics is just like studying herbs.
Yeah. I'm sorry you had that experience, first of all. And what it boils down to is our education in medical school. And truthfully, we're not taught much in the way of nutrition, and we're not taught much in the way of microbiome. And so that bias carries its way through folks' careers, and absent a curious mind and proclivity to learn a lot of this stuff independently, it's not part of most physicians' repertoire. And so the frustration that you experience on the patient end, as it turns out, is also frustration on the physician's end, because they don't know how to advise. And it's unfortunate. It's something that needs to change. I'm involved in the training program at the University of Washington, and try to bring the fellows up to speed and help to write the microbiome syllabus for the medical students. But yeah, it might take a little time. And in the meantime, my apologies.
Thank you.
Yeah, I'm involved in a similar training endeavor where you're a UW. I help with the WAMI side.
Oh, nice. Yeah.
Yeah. My mentor is a faculty member there, and so once every few months, I go and give a lecture on this, and the goal is really to just it's a special section, right? It's an elective. The goal is to get the students to be aware, so when they do go into practice, right, they already have some fundamental knowledge and some exposure to it. And because, just as Dr. Damman mentioned, this is really an education issue. Another part of it, though, goes back to some of the conversations we have in our industry meetings, which is the reliability of the data that we put out in the microbiome sector. And so until there is a little bit more trust that the data we are reporting is reliable and reproducible, I think there will still be some practitioners that will be reluctant to fully jump on board.
Yeah, I agree. And I would add that Scott and I teach on this in the integrative medicine programs. So the people who are coming back for education in integrative medicine are getting it. And that's another mechanism, right? Because you're working on the pipeline, which is absolutely where to start, but people who are already out there need this education as well. So we've got continuing education, we've got degree programs like our integrative medicine program. So we are out of time. I did want to give everyone a chance to do one last closing remark. So I'll just go in the order that we presented. So Scott, you are first. One last closing remark.
I wanted to mention something, and this is a bit anecdotal. a couple of years, I don't know if we had mentioned it earlier, but Leigh and I are not only colleagues, but partners in life. Leigh is my wife. And a couple of years ago, we did a plant-based diet for a week, just plant-based for a week. And at the end of that, we did one of the direct-to-consumer at-home gut microbiome tests. And we sent out our fecal sample. We got the report back. And something that I remember that stood out is that in the report, they said your diet is too carnivorous and you need to cut out the animal-based protein and eat
Hilarious.
This was after a week-long plant-based diet. So, just consider that in terms of the reliability. And I don't mean to, slander any company or doing it wrong. That's just the state of the field we are in right now. That's the best we can do right now. So, we're progressing in the right direction, but, it takes time. Rome wasn't built overnight.
Exactly. Yvonne.
Yeah, I feel like Scott took the words out of my mouth. That I was just going to again say, Rome wasn't built in a day or overnight, and we really need to give this time. But also, I personally, I love the microbiome, passionate about it, spent my entire career, not entire, some part of my career studying it, and I hope to stay in it forever, right? But I do, and because I love it, like a mom that loves a child, I can see the weaknesses and I can try to point it out and do my bit to fix it. And that's kind of where we are at in the industry. So I will say that we should be expectant for, I think, advancements, especially towards causation and not just still doing correlation studies. I think we should be expectant for more along the therapeutics line. And then one area that I'm particularly excited about is diagnostic potential or predictive potential, predictors of long-term health, that potential to use the gut microbiome profiling as a way to do that. Of course, once we work out a lot of the kinks with method variability, I think it has this powerful way of being able to help support holistic and integrative healthcare, because if there's anything in the body that is connected to everything, it's the microbiome. I think very few, maybe the immune system is the other one, but most other systems in the body don't have the far-reaching impacts the gut microbiome does, and it's powerful. So I'm looking forward to the next few years with excitement for sure.
Thank you. Chris.
Yeah, so the sentiment that I want to express is gratitude. Gratitude to institutes like Integrative Medicine at George Washington that are actually leading the charge in this education, as evidenced by the Symposium, and filling a niche, so to speak, to borrow an analogy in education. So yeah, thank you to Leigh and to my co-speakers and to George Washington and frankly everyone that has stayed to the bitter end here, and may this flame that's kindled here today be carried on and grow bigger.
Yeah.
Oh, wonderful. Thank you. That makes my heart feel so good. I echo all of those sentiments, especially the sentiment about maybe feeling a little bit frustrated that we aren't there yet, but knowing that we will get there, and that's just how science works. It takes time. But hopefully things will be moving a little bit faster, right? Over time, we're getting to know more and things will really ramp up. I think this is an absolutely super exciting place to be. Like Yvonne said, it touches everything, right? How many things can you alter, and it changes both, your cognitive capability, your, ability to lose weight, and what your mood is like, and how you digest it. It's so promising that once we do have a good understanding of it, I see it as being a huge area for therapeutics. With everything else, right? This is not going to fix everything by itself, but this is one of the tools in our toolkits when we're doing a holistic look at someone that we can utilize, a lever we can pull. And so with that, I will just close us off and thank everyone for joining us. It seems like you got as much out of it as we did from the comments in the chat, which I love. And if this is something you're interested in hearing more in, we would love to get feedback, because I'm sure this group would love to get back together and chit chat again, even if it was just for an hour. Thank you all for coming. I couldn't have done it without you.
Thank you.
Thanks, everybody. It was a pleasure.
Thanks all.